this episode of Action Hour, I'm back with microbiologist Linda Rhodes to discuss what she has learned as a citizen scientist monitoring bull kelp in the nearshore marine environment off the coast of Whidbey and Camano Islands with the Island County Marine Resources Committee. Take a listen. Let's shift the conversation to the bull kelp studies on Whidbey Island that you've been doing with the Marine Resources Committee. And I'd love to, kind of ask you some questions about the Marine Resources Committee first, to be like, what is it? How did it form? And how did you get involved?
Okay. So the Marine Resources Committee, or the MRC, is an advisory committee to the Island County Board of Commissioners. And it was established under the auspices of the WSU Extension. And I can't remember how far back that was. Maybe, don't quote me on the number, on the dates. I think that's fine. You can retrieve that information. But actually, it was established because Jack Metcalf and Patty Murray, our representatives to back in the 90s, I think it was the mid 90s, actually ensured appropriation of funds to establish something called the Northwest Straits Commission. And that covers 7 counties in the northern part of Puget Sound, or our part of the Salish Sea. And for each one of those counties to establish those MRCs. And the idea was, because those counties had so much shoreline and so much seawater adjacent to them, that it would be important to understand those resources and what that meant to the economy and the health of those counties, and to provide that source of information to not only to the county commissioners, but also to the planning departments and to develop the departments that are involved, county departments that are involved in shoreline development. So that since about 2012 in Island County, the MRC migrated from the WSU Extension being its fiduciary agent over to Island County Department of Natural Resources, which had been established in the early 2000s. And at that point, the MRC interacts very heavily with DNR, Island County DNR. And there is actually one of the county commissioners sits in as an ex officio member of the MRC. So there's a direct connection with some of the decision making that goes on in Island County. And because the county's departments don't really have the resources to do a lot of investigative work. They're there to do a lot of decision making and regulatory stuff, but they do need to have information. They, in some ways, do accept information that the MRC can come up with. And the bull kelp project actually came from an international effort, both especially on the Canadian side as well as on the U.S. side, to improve our knowledge about bull kelp. Eelgrass has been in the public eye now for probably 30 years. There's been a lot invested in understanding, what does eelgrass do? How does it function? Where is it? Are we getting more? Are we getting less? That sort of thing. Bull kelp, less so. Eelgrass is more intertidal or shallow subtidal area. Bull kelp is really definitely a subtidal organism. And
can you explain what subtidal means?
The bottom never goes dry. Never when the tide goes out, even on the lowest tide that you have, you'll never see that. It's never exposed to the air, to the atmosphere.
Oh, okay.
Yeah. So there had been efforts in Canada. Canada had research groups in Canada had recognized some kelp loss, but really the one of the big things that happened was in California. In 2000, there was sort of a series of events that happened that resulted in California losing, and some of the numbers sound incredible, but up to 90% of their kelp along the shoreline. And I don't know if that means acreage or, but they lost a lot. And it was not just a single event. And they're down there, they're talking about not bull kelp, but macrocystis, I can't remember what the common name is, but it's a different kind of kelp. And we have that here in Washington State, out along the Straits of Juan de Fuca. There's macrocystis that comes in, and there's a zone of overlap where nereocystis or bull kelp and macrocystis are together. And then as you move further in, it just becomes bull kelp. So, but in California, they had a big harmful algal bloom back around 2011. It affected invertebrates that's like sea stars. And then there was in 2013 and 14, there was sea star wasting disease all up and down the coast from Alaska down to Baja California, just wiped out sea stars. So sea stars kind of got the double whammy on them. And so the numbers of sea stars plummeted. And then in 2014, we had the blob that started. In 2015, we had a big El Nino event and the sea surface temperatures rose. So with the drop in sea stars, rise in urchins, because sea stars prey on urchins, and then those water temperature changes, that's when California Department of Fish and Wildlife began to see that there was just huge losses in kelp. And I think they're still working on a restoration. But that was kind of a signal of, okay, maybe we better start worrying about it. Things look pretty good around here. Maybe this is something that the Northwest Straits Initiative took up the cause of saying, can we set up a citizen science monitoring program? Because there's no money for professional scientists to do that. And that's where the MRCs all came in. So all 7 of the MRCs set up monitoring projects while we were learning how to set things up, because this was all totally new. And it was going to be boat-based, that is kayaks. And we were going to use citizen scientists for it to do the monitoring. And I'll say that.
Go ahead.
No.
Oh, I was going to ask, like, what are the kind of qualifications to become a citizen scientist, a part of this project?
Well, you do need to coordinate with the MRC in order to be a participant in that. Sound Water Stewards has been a great resource in terms of people who are interested in doing this. Need a boat. You need to get some training and, demonstrate that you're seaworthy, that, you know what you're doing in a boat and you're not going to drown yourself. Need to get some training about how to use a handheld GPS, which will actually be used to track the perimeter of the kelp. And you need to coordinate and go out with team leads. Each one of the beds here in Island County have got a team lead who coordinates when they're going to go out. And you need to go out at a specific time. There's certain tide heights that they want you to conduct the survey at. And you can do it on a monthly basis, or in some cases, we have beds that we know a lot about their monthly growth pattern. So we just do that once, because it turns out that Washington Department of Natural Resources, and Helen Berry specifically, who leads this effort, uses the maximum area of the bed for each year as a way of tracking year-to-year changes.
Cool. I Googled bull kelp, for this, and I've seen bull kelp a lot on the beaches, but it's so funny because they're huge, long, like whips of kelp. And I'm wondering if you could kind of describe for people listening what bull kelp is, both what it looks like and then what its purpose is, and, why it matters that it's here.
Sure. Well, first of all, it's an algae, so it's not really a plant, although even people that are professional kelp researchers will call it a plant. It has an attachment point down at the bottom, usually attached to a rock. It's called a hold fast. People say, oh, it's the roots. Well, not really. It's just a way of holding on to something so it doesn't travel all over the place. It has a long, skinny tube that ends in a rounded bulb that's actually got carbon monoxide in it for buoyancy. It synthesizes carbon monoxide. And then at the top of that bulb comes these long blades, flat blades that, again, people like to think of them as leaves. And those blades grow continuously during the growing season. So the tips of the blades slough off and become food. The actual kelp, it is a brown algae, and it uses photosynthesis to grow, but it can also absorb nutrients directly, dissolved inorganic nutrients from the water. So it can absorb certain nitrogen and phosphorus compounds directly from the water. It actually is, you only see it certain times of the year because it's an annual. It grows up. And then at the end of the season, by the time we get to October and November and we start getting storms, those guys will get churned up and torn up and either released as mats that float out to sea, or it could sink and go down to the bottom and be fertilizer or serve as a nutrient source itself. It has a 2-stage life cycle. So you know how many plants might have 2 different forms during their life. That's true for bull kelp. So there's the big blades that you see. If you were able to look closely at it, you'd see there's a lighter colored area in the middle of them, and that's a place where it produces little microscopic organisms called zoospores. And those guys have tails on them. And at the right time, those zoospores get released from the bull kelp blades and they will swim down. I think they have like 48 hours to find a place in the bottom to hunker down and settle in. And from those zoospores, they produce a little tiny plant that's only a couple organism that's only a couple millimeters in size. That's called a gametophyte. And these are the sexual forms. So there will be male and female forms. And those guys grow up and they'll release their gametes, which merge and become a zygote. That is a 2N. So now if you're talking about DNA, the zoospores have a single complement of DNA, and the gametophytes have one N, but when their gametes fuse together and make the zygote, that's 2N, like us, 2N. And then that little zygote now has to settle down someplace, and it's what grows into that huge organism that you see with the long blade. So it has to do all of that in an annual cycle. And that's So it really, a kelp bed, when you see a kelp bed, you're really relying on the organisms that are there to be producing the next year's crop. But it has to go through that gametophyte stage and then zygote stage in order to grow. The sporophyte, the big thing that you see, I mean, in Alaska where they have long daylight, I think they've measured it growing like sometimes 3 feet in a day. I mean, that's a phenomenal, you'd think you could just sit there and watch it grow. But that just shows you the role of photosynthesis is also important for their growth.
And so the sporophyte is the big, long, tube that we see. And before it washes up on shore, you said it's attached to a rock somewhere under, like on the ocean floor or on the sea floor. Right. Okay. So that's where it's hanging out. And what is it doing when it's on the ocean floor? Right.
Okay. So back in, these sporophytes probably start growing, maybe in March or so, and they're growing longer and longer. When they get up to a certain height, they can start providing some physical cover for small fish to hang out in. And it can also provide invertebrates, little like sea slugs or not sea slugs, but sea snails can attach to the surface, and they can actually eat the goo that's produced at the surface. Because if you hold bull kelp, you'll feel it's very slimy. And that's a food source for little guys that scrape and eat that kind of material. You can see there's bryozoans. Those are another kind of invertebrate, especially later in the year with an older organism. You'll see these little round white circles on them, and that's another kind of invertebrate that sets out. So it's called biogenic habitat. That is, it's biological. It's producing its own habitat by the size that it is. And people talk about kelp forests, right? Sort of, again, going back to a terrestrial analogy, but it's providing shelter and it's providing food to a lot of different kinds of organisms. I know we've been in the bed and seen lots of herring schools, small herring schools hanging around in there. Because again, once you start feeding something, other things come to feed on it. And you sort of get this food web developing that uses that space, that biogenic habitat to grow. And it's only there for a certain time of the year. And it's kind of a nursery area, if you want to say that, because juvenile rockfish hang out in there, probably down at Possession Point, where there are a lot of rockfish, the adult rockfish that spend time on those overhanging areas that are just north of it. I know we've seen certainly herring and sand lance in there. I've seen other kinds of groups of small fish. So that's the thing. We don't see very many big fish in there. They're all small fish. We've seen big crabzoea. So they just look like little dots, just big collections of crabzoea. So I would say it serves as kind of a nursery area for a lot of marine critters.
That's really interesting. And because it's perennial and it's only there for part of the year, does it seem like it's kind of its timing of its habitat creation is in a timing that is with the life cycle, like a particular stage of the life cycle of certain other animals?
I'm sure it is. I don't know that for a fact.
But I'm just wondering. That's a curiosity that I have, to be like, do the herring know? Right? Do the little right. Do they know that a certain time of year the quote kelp forest is going to be there?
Yeah.
And that's curious to me. I want to go back and ask a general question about the difference between a plant and algae. What makes algae?
Oh, I'm not sure at this point. I know they're considered protists, but I don't know what all the characteristics. The kelp are considered protists, and they're actually microscopic protists that never get as big as this either. But I honestly, at this point, don't really know exactly what makes a protist versus a plant. It's probably some taxonomic features that I am ignorant of.
Okay, totally cool. So another thing that I'm interested in, so we've talked about how this bull kelp creates a perennial habitat for.
It's an annual. It's not a perennial.
It's an annual. Sorry.
Yeah.
It's an annual, and it creates an annual habitat for different species and functions as part of a food web, like kind of creates a food web. I know that people have also said that it is helpful in the reduction of carbon. And I'm wondering if you know about that at all.
Yeah. Well, it's certainly because it's photosynthetic, right? And it can incorporate and it can actually pull carbon out of the water. And so that means it is doing that. But, the thing about carbon sequestration is you have to think about time scales, right? So because it is an annual, at some point it is going to degrade, and bacteria are going to be responsible for that, probably, most likely.
Yeah.
And that's going to produce CO2. So it can do carbon sequestration in a localized short-term perspective. I don't know about long-term, to solve our high CO2 levels or, carbon in the ocean. I don't know. I don't think that's got the right characteristics to address that particular issue. Yeah.
Yeah. I'm wondering what exactly is the MRC monitoring right now as it relates to the bull kelp? What is the kind of what are kind of the characteristics that you're looking for? Yes, on an annual basis, and then what are the trends that you all are seeing?
Right. So early on, because and we didn't really know anything about bull kelp, and not many other people knew much. I mean, again, Helen Berry knew quite a bit about it, but she's one person, and bull kelp wasn't the only thing she was involved in. So what we were trying to do is collect information that would be useful for her to be able to have eyes on a large part of Puget Sound, particularly up in this northern area, because a lot of the bull kelp mass in Puget Sound and the Salish Sea resides in the Northwest Straits Mission area. Yeah. I mean, there was a survey that was done by somebody back in 1911 for a totally different purpose. They were thinking about trying to make gunpowder and using it as a source of material for that. And then some other ideas about trying to use it for fertilizer. But there's a survey done back in 1911. It was like the overwhelming majority of the biomass was actually up here along the Strait of Juan de Fuca, actually Smith and Minor Island and Island County bull kelp. It represented a huge proportion of it. So we know what historically it was like before serious industrialization was happening in Puget Sound. Now, again, Helen's one person, and so the idea is, can we give her information? Can we provide information that would help her develop structured studies that would allow her to understand the importance of kelp in the shoreline? So our main variables that data's collected on initially was, what are the growth patterns across the seasons for the bull kelp beds? And then we started taking some like surface temperature measurements just so that we would have some kind of temperature data, and what are the depths of the beds. So those were kind of the main things that we collected information on. And it turned out for Island County, because of our kind of unique position that we have totally marine water on one side of Whidbey Island, and then we have this heavily river-influenced body of water between Whidbey and Camano Island, but there are definitely bull kelp beds in both locations. So that means that the ones that are getting a lot of freshwater input are actually able to endure those kind of conditions. So we chose one bed, Eby's Landing, that faces out to the strait in Admiralty Inlet. We have Paulnell Point, which is, just about less than 4 miles away from the South Fork of the Skagit. And then Lowell Point, which is Camano Island, the south end of Camano Island. And then Possession Point, which is another big bed, big fishing area, and actually is where that river and marine waters kind of converge, come together before they go to the South Sound. And conversely, when the tide's going out, all that water from Central Sound, the heavily industrialized and urbanized influenced waters, are coming up. So we felt like those represented some good locations, and have been able to sort of maintain monitoring there. I think Hoypus Point, that's near, and the reason why we kind of focused on the east side of the Whidbey Island is when you look at the Skagit River, which is where the biggest runs of wild Chinook salmon come from the Skagit. One of the questions is, does this provide any kind of protection or cover for juvenile salmonids as they come out? And, half of them go south and the other half go north, and they would have to go around Hoypus Point in order to go out Deception Pass. So Hoypus it's not a very big bed, but, kind of interest if they're going to pull over. We know that salmon go around the corner and hang out and wait for the tide. The question was, do they actually use that bull kelp bed? So what we discovered is that the kelp that's on the side that gets a lot of marine influence, at least the Eby's bed, that bed shows up early, and it shows up pretty strong. And in fact, we view that as kind of a control site. It's like, if we start seeing things going downhill there's something bad happening. Something serious is changing, seriously changing. Possession tended to be a little bit slower in coming up to the surface, but some years it can be early, some years it can be late. Mm-hmm. The beds that are on the Saratoga Pass side between Whidbey Island and Camano Island, they are very slow to show up. They do show up eventually, but it might be the maximum area. It might be September before you see the maximum area, whereas with EB and typically with possession, you'd see it in August. Mm. So I think that those beds in Saratoga Pass, there's something that not hinders, but it delays the sporophyte growth. Mm-hmm. I do wonder about turbidity because rivers carry a lot of sediment with them. Mm-hmm. And there's different ways to generate turbidity. You can have sediment, chemicals or particles that actually block light, or you can actually have algal growth. Sometimes, microalgae will grow, and everybody thinks about harmful algal blooms because they're microscopic and they can get very dense. But you think about just growth, planktonic growth can get very dense and can also block light. So there's probably some potential to look at what the influence of turbidity and light blocking characteristics might be along there that might explain why that takes a longer time for those Saratoga beds to show up. Mm. Collecting temperature data, again, we're talking about citizen scientists in kayaks, so you can't be using super expensive equipment to deploy things, and the risk of losing stuff, you always worry about that. But Canadian work had really shown that 17 degrees centigrade seemed to be a critical temperature for the zoospores. So you remember you've got the kelp blades, the mm-hmm. sori that the zoospores come from. And if those guys experience temperatures of 17 degrees for, I don't know for how long. I'm trying to remember how long their experiment was, but it was maybe for a day. It actually compromises their ability to swim, and many of them can't swim. And so fewer of them successfully get down and establish themselves in the bed. So obviously with temperature rise and the heat blobs, you worry marine heat domes, you worry about, temperature being a factor. Mm-hmm. Many of these sori, actually, we have seen sori that have released underwater. So it means it's not just the stuff that's at the surface that may be getting temperatures further down. And we also don't know what kind of influence temperature has when either the gametophytes, those little tiny guys that are at the bottom, or when the sporophytes are young, do they have some kind of a temperature constraint? So one thing we expanded into was trying to get temperature profiles using a handheld temperature and salinity device to understand that. So those are kind of, and I know I was the project lead from the start back in 2015 up to 2020. And then I kind of turned it over, although we're still monitoring a couple of beds. I know the current project leads are very interested in developing much more complex sampling technologies to look at more temperature and salinity and a lot of other things in addition. Mm-hmm. One thing I focused on early on was people aren't going to care about kelp if they don't see pictures of them, if they don't see videos, right? Because that would be the question. Why would I care about kelp? I can see it from the shore, big deal. So we focused on taking underwater videos from the kayak. I'm not a diver. Mm-hmm. But we've got GoPro cameras. We had drop cameras that we could lower down and look. And the thing is, there is a lot of submerged aquatic vegetation. There is truly a whole meadow of other kinds of kelp and other algae growing underneath all of this kelp. So it isn't just these long, skinny things sticking up and nothing there. There are areas like that, but there's all this other submerged aquatic vegetation that we don't know a whole lot about because it's not readily accessible, like eelgrass. Mm-hmm. And it's clear that that is part of that biogenic habitat that kelp provides.
Wow, that's very, very cool. I do think that it'll be interesting to see if the MRC can gather specific data sets around how temperature affects the different parts of the life cycle of this plant, or not plant, algae. Just because I do think that sea temperature rise is one of those things that, ocean scientists have been looking at for decades now, just to see how it is impacting growth in general, like what are the limits for different species in their life cycle. And, if you get a flash, like a heat flash during a very specific time in the life cycle of an organism, it could be detrimental to that organism. I know that
Absolutely.
You mentioned earlier, the sea star wasting disease, and I know that impacted populations near Deception Pass on Whidbey Island. And I think that there is, I went to a Sound Water Stewards event, and someone asked a question about that. And I think that, the scientist who was given the presentation, I can't remember his name right now, but he had mentioned that they are still looking also, in those areas to see if there's recovery, and that it's very slow, that there is some, but not a lot of recovery in that area. And so this kind of gets to the question that I have about conservation, right? Conservation versus restoration. And, the importance of that. Cause I know when we met and we spoke, you were talking about how important it is for Island County and for our community to be conscious of bull kelp and be in the mindset of conserving the populations we have versus being in the unfortunate circumstance and the expensive circumstance of restoring these habitats. And so if you could speak to that a little bit, that would be great.
Yeah. Well, having done research throughout Puget Sound since the 70s, the late 70s, and a lot of the research focused on Central Sound, the urbanized areas in South Sound, and some of it in Hood Canal. What I see here in the Whidbey Basin is that we have probably some of the better nearshore habitat that's available in Puget Sound. Now, it's not the same as, the Strait of Juan de Fuca along. They have fantastic shoreline habitat, but it's very different from ours. This again, when you think about if you're going to be focused on salmon, for instance, this is where salmon need to grow up and grow strong before they cross out and hit the road and head out to Alaska and the Gulf of Alaska. And so, and our shoreline is a place that they can use to get the growth and development that they need safely. I mean, that's the thing. They need to get big. Survival is often a race to size to get big enough. And so we have some really fantastic shoreline here. We have some amazing resources like the Skagit River and the animals coming out of the Skagit River and the Snohomish River. So, and we have a lot of rockfish. So people really need to understand that what we need to conserve here is not just a pretty shoreline or a nice view, but that we have important habitat that's not replicated anywhere else. There is not a whole lot of redundancy of the kind of shoreline that we have and that near shore habitat. And so, and the thing about if we lose bull kelp, right now, we don't have the tools to do the restoration. The Puget Sound Restoration Fund, the PSRF, they have they have one guy, Brian Allen, who is an expert. He's figured out a lot about how to grow. But this is like the bacterial thing, right? In order to understand what a bacterium grows, you got to culture it, and then you have to subject it to experimental stuff to see what it does. Same thing here. You have to be able to grow it in order to understand what it needs and what it can and cannot tolerate. Well, we're not at that point yet. And so they are working now. PSRF is leading the effort to figure out how can we plant bull kelp to restore an area. However, it would be good to understand why the bull kelp was lost in the first place. If it was there and then it went away, do we understand that? Because you could go out and put bull kelp out there and it would just suffer the same fate of dying. So there's so much we don't know about bull kelp that if we lost the beds that we see around Island County right now, that's it. I mean, we can do a lot of hand wringing and maybe try to, after the fact, diagnoses of what caused that loss, perhaps, but observation is not a very efficient tool for understanding cause and effect relationships. It can give you clues about what we should be paying attention to, but experiments are what really demonstrate a cause and effect relationship. So if temperature is an important feature at a certain stage of their growth, we're probably not going to figure that out by doing observations like what we're doing with the monitoring, the MRC monitoring. That needs to be done in an experimental setting. On the other hand, if we could, this monitoring provides us with an ability to start seeing patterns that are occurring. And here's a good example. We do Eby's Landing. We've done 10 years. We've gone out there and done this monitoring. And I always felt like that's a pretty robust bed and it's a control site. We don't need to worry about it. But last year we went out and we noticed that there was a big empty spot. We called it the donut hole, the big empty spot. And we actually, traced that, made a track for that so that we could, when we put it up on the map, we could see where that donut hole was. This year we went out and there was a lot less kelp at that end. And when we superimposed last year's donut hole on this year's outline, the donut hole is now outside of that margin. So all of the kelp that had been around the donut hole and everything south of it didn't show up this year. And I don't know why we didn't have time to do the drop camera. That's what we've done in the past. Whenever we've seen a decline in a particular area, we'll take the drop camera to look down and go, are there sporophytes growing up, but they're just not at the surface yet? Is it just because we don't see them at the surface, or is there truly nothing there? And we didn't have time to do that. We just did that earlier this week. But I'd like to know, we'll probably go back out in September to see, well, if there was something down there, did it show up? Same thing happened in Paulnell in 2021. The area of the bed was half what it had been the other 7 years before that we did it. And there we did go back out and did a couple extra surveys. We put the drop camera down and looked around, and there were no sporophytes. I mean, the fact that there was nothing at the surface indicated there was nothing at the bottom. Now, the good news, we went back out in 2022, or maybe now I'm getting mixed up. Maybe it was 2022. It was half, and then last year we went. Yeah, that's what it was. It was 2022. It was half that area. 2023, when we went out, it was all back to what it had looked like before 2022. That suggests that something happened. We don't know what happened, but there were enough sporophytes producing zoospores to repopulate those areas that didn't have anything that's there. So there's these natural fluxes that maybe occur, might not have anything to do with temperature or turbidity or what. We just don't know. And that's the kind of thing you need to understand. Maybe they have cycles that are 10-year cycles.
Yeah.
And that's where you need this kind of information.
Yeah, it's interesting. You mentioned observation and then, testing, right? Like observation being a good tool for pattern recognition and testing being a good tool for cause and effect relationship. Are there other scientific tools that would be helpful for this project? And from your point of view, how are there more resources that would make this more effective? like if I'm sure the answer is yes, right?
Yeah. Well, one of the things that I would love to see is there are a number of buoys that are deployed by the University of Washington, particularly in Hood Canal, because it experiences those hypoxic events. And so they have some very sophisticated equipment where they actually take profile data. They have a little crawler on the buoy that goes down and up, and it'll collect, all the parameters that you would be interested in, and it does it year round. I would love to, maybe not, those are called orca buoys, another use of the word orca. I would love to see some kind of year-round equipment deployed that could give us a kind of temperature profile, temperature, salinity, turbidity, that kind of information. We did manage to get one year when I worked for NOAA, I had a couple of divers stick there's these little things called hobo recorders, and they run off of a little tiny battery, and they can go for 3 years. They can collect, data pretty frequently. And so we actually did bottom profiles down at Possession Point for a full year. So we could actually see, it was so exciting. And it was just a very simple piece of equipment that was down at the bottom. And they retrieved it and brought it back. And I'm like, wow, imagine if we could get this kind of information in all of our beds, and not just at the bottom, but maybe at some mid-water point where, the sporophytes are starting to grow up. That would be really useful. And that's possibly within the realm of MRCs, but probably not. That probably, because it does require some divers to be involved. Possibly DNR, but again, they don't have a big budget, and that's a limitation. But, having those kind of recording devices year round would be huge.
Yeah.
The other thing is, if you're interested in turbidity or understanding that, collecting, there are these traps, sediment traps, that actually collect material and tell you how quickly and what kind of material is collected. But I think those year-round recording devices, that'd be the one thing that I would love to see, and I think would be extremely valuable information.
Yeah, I agree. That would be really smart, and maybe something that could be funded in the future. I think if the county and the Northwest Straits Commission is able to invest in monitoring, in these, as you said, like these areas that have very special nearshore habitat, then Whidbey Island could become. It already is, but could become a better resource for.
And what's amazing is we don't really have that many kinds of autonomous monitoring devices in the Whidbey Basin. They got them all down there in South Sound and all these other places, but we don't have that. I think there's a college in Everett that is tried to do that, and I think the Tulalips have tried to have a few buoys around the areas near their areas that they're concerned about. But some of these we got nothing. They send the Prism cruises up here. A big ship comes up and does water grabs, but that's just one time. We're not seeing that total cycle.
Yeah, seeing the total cycle would be really helpful. And that kind of speaks to the kind of sense of, or the observation that we have a very special habitat, but we don't have a lot of information about that habitat in, overall, right? Like there's, that's interesting. And I know that we've, something that I wanted to ask you about in terms of pointing like scientific study in a particular direction, and the role that intuition plays in scientific exploration, because you have to kind of be journeying into the unknown, right? Like oftentimes when scientists decide they're going to start collecting data, I'm curious, how do you know where to start? And what's kind of the role of intuition in that process?
Yeah. It's incumbent on a scientist to understand what they're studying, in the sense that becoming familiar with it. Okay. So for me, I knew nothing about bull kelp at all. I started all of this, and I thought, oh, okay, well, I'll learn something and maybe contribute some useful information. And then, having done some of these beds for 9-10 years, there's a familiarity with it, right? I'm starting to go, oh yeah, we know. Remember last year? You start to understand. It's like going into a forest. I know people have favorite hikes, and they go someplace repeatedly, and then they start to know. They know the individual trees, and they know what time of year it is and which birds they might expect to see. And so I don't know if you've ever heard of a woman named Barbara McClintock. She had done work on corn back in the 30s, and she was involved in understanding why corn kernels might have different colors. And it turned out she actually discovered that genes could be mobilized from one cell to another, called jumping genes. She got a Nobel Prize for it. But the important thing, that seems kind of obtuse, but the thing is that she wrote a great book called A Feeling for the Organism. And that's where that came from. You're talking about intuition, and what it is you're working with these less linear thinking, less parameterized things that you maybe don't put a number on, but your brain is very good at integrating a lot of different kinds of information to come to an understanding about what you were studying. And I think that's what you need to have. So, doing citizen science is great. Probably the best feature of it is awareness. It spreads the gospel, so to speak. People go, oh, I never thought about that stuff before, and so it's good to have people invested in it. Mm-hmm. But I think there's also a territory where citizen scientists can really understand something. And even a professional scientist may not be able to do that because they're busy dealing with grants, developing structured studies that can get approved and produce a product that can inform policy. So in many ways, they're being scientists, but they don't have that time to go out there and just kind of sit in the bed or go there repeatedly and start to understand what you think is going on there, or just to observe it and absorb that and synthesize. It helps to synthesize your perspective and help feed that intuitive choice. Mm-hmm. So I know that like doing transects is a popular tool for collecting data. So transect is you go through and you say, I'm going to, set up a square or put a ruler out, and every 2 feet, I'm going to collect some kind of piece of information. So it's kind of a very randomized way of collecting data. Mm-hmm. And that's good when you don't know anything, But once you start getting familiar with it, you go, okay, how can I use a transect in a more creative way? Or how can I direct what I'm doing to get more information from it? So, I kind of regret last year not going out to that donut hole again and looking around with the camera, because we knew that normally we'll see kind of thin spots, and we're going, okay, we know over the years it fluctuates, but that, we knew that something was going on, and we just did not follow up with the drop camera. And that would be, so as opposed to using a drop camera where you just go through on a transect, right? Now you take your camera and you go over like, oh, this is weird. We haven't seen this before. Let's put the drop camera down here.
Yeah. And your intuition would tell you, like, there's something different. Let's explore. Let's go look at it. Mm-hmm. Let's go find out what's different. Just like, and I appreciate the feeling into the organism or feeling into the habitat and how presence and being there, right? Like whether it being out in the field or being in nature in general, that if you go back to a place over and over again, you build a relationship with it, you observe over time, and you get a sense of what it's like. And I think that what I find, I think what I'm gathering from this conversation about monitoring bull kelp is by monitoring bull kelp and having that as the starting point, that's an opportunity to observe an entire habitat. And that's giving citizen scientists information about the nature of that habitat and how special that habitat is. And also, what you said in terms of salmon, and Whidbey Island kind of being a place for salmon to grow, like the habitat, nearshore habitat, it gives me this overwhelming or overarching sense that Whidbey Island is a nursery. Right. That the nearshore habitats of Whidbey Island might be a very critical and crucial nursery for lots of different species of organisms that we haven't had the time or the energy or the resources to explore. And so intuitively, I say, I think we should be thinking about that and putting resources towards that. And, studying bull kelp and expanding, the monitoring systems, and also the opportunity for testing to kind of understand, the importance of what we have, right? My dad always says this. He's like, I'm just trying to understand what it is that we have. And he said that because, our family has acreage in Coopville, and he spends every day walking through the woods every single day for hours. And he knows his woods like the back of his hand, and he learns a new thing every day about it. But he always says, I'm just trying to understand what we have. And I think that that's kind of where I think of in terms of like, why is conservation important? It's because we haven't yet understood what we have. And so you have to keep what you have in order to understand its importance and its significance. And from an ecosystem standpoint, its function, right? We don't understand the function of our nearshore environment in its entirety. You know, we don't really understand it. And yet as human beings, we often have the hubris to manipulate it without understanding.
Oh, yeah. Well, that's because society has prioritized other things as being more important, right? I mean, that's the problem is, so, and just to give you kind of a cool example, this Paulnell Point site, it's very different from the EB site when you look underwater because it's got tons of, I've yet to see herring over there. I'll have to admit, we haven't seen baby salmon. But
I was going to ask you.
Yeah. You know, I think those that the kelp bed comes up maybe a little bit late for salmon because salmon tend to start moving out April and it can dribble on through the entire summer. So it might provide some of them. That's the natural spawn guys. The hatcheries, they come blowing out in June. And I don't know, we hadn't seen a salmon out there in June, but remember I told you that they're slow to grow. So they're not very tall at that point. But there are tons of shiner perch over there. I mean, that whole bed is full of shiner perch, sea stars, and we haven't done an intensive survey, over time. We didn't see later on. We didn't know. I didn't have the drop camera going then, but we haven't seen signs of sea star wasting over there. But we kind of started using the camera after the peak of that epidemic occurred. Lots of sea stars, lots of sea cucumbers. It's amazing how many sea cucumbers there are over there. Lots of crab. The folks that live along there, they love to go crabbing there. They know that that's a reliable place to pick up crab. So it has a very different kind of vertebrate or invertebrate collection of animals compared to the ones out in EB that's serving, like you said, as a nursery. That's probably also serving as a nursery. A lot of things eat shiner perch. They're at the bottom of the food web for a lot of the birds, and we do see and seals. Seals like to hang out over there. So it's a very different place than EB, and it's kind of a thrill to go, oh wow! So now you see it's a bull kelp bed, but not one size fits all. The description is that fit all of them. They have different functions depending on where they're located. Yeah. And the other thing I want to point out is, we don't think very much about our connectivity to the mainland, which is conveyed by the rivers. And that's where that I think is a link that is worthy of being investigated to see, how much of the sediments, because that's, the Skagit's a managed river. In fact, what's interesting is I went back and looked at the gate, the Mount Vernon tide or river gate measurements, and the previous April, I think, or maybe it was a little bit earlier. Anyway, one of the biggest releases of water occurred before we saw that huge reduction in area. Now it was months before. It was like 4 or 5 months ahead of time. So, but you think if that brought enough sediment out to bury those little gametophytes or the little sporophytes just long enough that they didn't have a chance to break free and start to grow up. But it wasn't a long-lasting issue. The guys that did manage to come up repopulated the area so that it could have been something as simple as that, just a huge release of water and sediment. So that's a connection to the mainland. And so just because, it's not just what we do to our own shorelines, but what are the other influences that come in and affect it?
Yeah. I mean, that's one of the features of water, right? Is it flows and interconnects a lot of things, and and none of it is happening in isolation. Right. That's really interesting. I love that. And I guess a question, maybe one last question that I have is, based on the observations that the MRC has been doing around bull kelp habitat, have there been any observations or any trends that you've noticed about how human beings affect bull kelp habitat and our relationship to the habitats that you guys are studying?
I've not seen, I can't say that I have. Now, I don't do the possession point bed. We just did it the first year and then handed it off to somebody else. And I don't know, that's a bed that gets a lot of human traffic because you get the fishing season opens up late in the year. There are a lot of divers. They love diving along in there. So there could be some impact there just because of the presence of humans. Yeah. I don't know about runoff, from land and how does that affect bull kelp. So that's kind of an influence. None of the beds that we monitor, well, at the Hoypus Point, there was kind of a hardened shoreline, an old hardened shoreline nearby. And that would be a question to look at their data and see. And that got removed a couple of years ago. So that might be a good one to see. Did that hardened shoreline, removing that, did that have any kind of effect on that bed? That was one reason for choosing that bed. But yeah, at this point, I can't say that I've observed any kind of direct influences. I mean, aside, nobody wants to drive their boat through there because it'll clog up their water intake system and wrap their propeller. So unlike eelgrass, which some boaters seem to be very happy to do, like up at Coronet or up at the Deception Pass, Coronet Bay, there have been evidence of people driving through the eelgrass beds and leaving kind of a track there. But I don't think they'll take on bull kelp.
Yeah, bull kelp has a presence, a very commanding presence.
People go way out to avoid it.
Is there anything else that you think people would be interested in or that you want people to know?
Oh, just, learn more about something, about your natural habitat, whatever excites you. That's important. And having that observation, that repeated observation is something that is highly valuable. And you never know when that knowledge is going to be needed. That's the thing. So you should always do it for yourself. And you can be selfish and say, this is what I want to learn about. But just be observant and respectful.
Yeah, observant and respectful. Well, thank you on that note. Thank you so much, Linda, for speaking with me today and for sharing all of your knowledge. I really appreciate it.
Was.
Thank you.
All right. Well, thank you for asking, Amanda. I feel honored and privileged that you felt like I could share useful information. So,
oh, yeah. No, you're incredible. Can't wait to talk again.
Another man up in the pulpit in the State House. Head of the table with a black pen, scratching egos, talking too loud. Externalize every outcome, every warm gun, every profit. And kneeling down to the flashing numbers, the NASDAQ, the cold constructs. Running backwards, so just keep moving, just keep moving, just keep moving. Unlearning every lesson that our mothers tried to teach us.
This podcast is a production of the Whidbey Environmental Action Network and is made possible by our generous donors. Produced, edited, and mixed by me, Amanda Bullis. Special thanks to Marianne and Steve for their years of service through action and for lending their voices to this podcast. To WEEN's Executive Director, Marnie Jackson, and our board for their leadership. Special thanks to Whidbey musician and WEEN board member Nathaniel Talbot for letting us use his track, Intuition, from the album Animal, available on Bandcamp.
Every day on your paycheck, a volume of confidence against the idea of your body being put into your own hands. So please don't hold it, please don't hold it, please don't hold it back. Unleash the power, the straight logic of compassion. What I need now ain't that old religion or the cold hard facts. Just need a woman's intuition, get us on track. Just need a woman's intuition, get us on track. Stepping out of the shadow of a false man's allegory, slowly erasing that old violence from the story. Recoupling emotion, slow emotion to intelligence, granting the planters, healers, teachers, feelers a new relevance. What I need now ain't that old religion or the code